Welcome to the Syngenta Innovation Center, located in Research Triangle Park, North Carolina. This is our flagship R&D facility for Syngenta seeds. Named by Science magazine as the top agricultural employer, Syngenta has more than 6,000 scientists in over 100 countries transforming agriculture day in and day out. Our 500 world-class scientists at RTP use a range of cutting-edge techniques such as genomics, genetic modification, and digital technologies to develop new crop varieties with enhanced yield potential and are resistant to pests and diseases, allowing growers to feed the world in a more sustainable fashion. The original facility here at RTP was founded in 1984 by Dr. Mary Dell Chilton. Mary Dell was instrumental in developing a technique called transformation, the ability to introduce beneficial genes into crop plants, a process that is still foundational to the science and work completed on site today. She has been the recipient of many awards, including the World Food Prize and, more recently, the U.S. National Medal of Technology and Innovation, bestowed by President Biden in October 2023. Today, our 55-acre campus houses a state-of-the-art 375,000 square foot facility consisting of 4 floors of research laboratories and the Advanced Crop Lab, one of the most sophisticated greenhouses and growth room complexes in the world. With 1 acre under glass, 30 greenhouse bays, 21 growth rooms and 2 seed stores, the Crop Lab's sophisticated design independently controls the temperature, humidity and carbon dioxide levels, and plants benefit from automated irrigation and fertigation systems. Our biotech product development pipeline starts here at RTP with discovery sparked by farmer insights before moving through to early and late-stage research and then into field and safety evaluations, all prior to commercial product launch. New trait genes can come from a variety of sources, including soils and other environmental samples. With the advancement in genomics and computational technologies, there is a large amount of DNA sequence and structural biology data we leverage to discover new genes. More recently, machine learning-enabled computational biology has opened incremental ways to design novel traits. Modern automated approaches to gene handling and testing have been developed, expediting the trait discovery processes. Now we can screen thousands of samples in a high-throughput manner. Gene candidates are introduced into plants by agrobacterium-mediated transformation. Corn ears are collected from the crop lab, sterilized, and embryos isolated. Genes of interest are transferred into agrobacterium to infect maize embryos. Cells which take up the gene of interest are then regenerated back into a plant via a shooting and rooting step. Thousands of plants are generated by this transformation process. Next, a detailed molecular analysis is conducted using PCR, next-generation sequencing, and mass spec to select the desirable plants to place back into the crop lab for efficacy evaluation. To address the increasing number of insect traits in our pipeline, we have opened a 13,000 square foot insectary in late 2023 to rear economically important insect pests and to conduct insect bioassays to identify plants that are resistant to these insect pests. Additionally, we conduct efficacy testing for diseases such as Asian soy rust, which is a major issue for soybean production in South America. Plants which contain herbicide tolerance traits can be selected and progress to the field for evaluation. To complete the registration process to enable a new commercial product launch, a detailed, comprehensive data package is produced for regulatory authorities worldwide. A multitude of studies are required, and the regulatory package must demonstrate the equivalence of the biotech variety to its conventional material. Additionally, mandatory studies are conducted to highlight zero human or environmental safety concerns. Over the coming decade, our research and development pipeline is poised to bring new products to market, which benefit from revolutionary digital technologies, gene editing and biotech traits. Here at the RTP Innovation Centre, we are fueled by accelerating farmers' success, and we live the Syngenta commitment to innovation, collaboration and partnership with farmers.